So the first thing I'd like to introduce is what are the ClinGen goals as it relates to data access in the software products. So I'd like to provide a brief introduction to ClinGen as it relates to software and data for those folks that aren't quite as familiar or outside of the ClinGen group. Next, I'd like to talk about where ClinGen data comes from and where it resides. How do we access ClinGen data through ClinGen software? And I'd like to focus on two particular products as it relates to APIs and streaming. The first one would be the ClinGen Allele Registry, and do overview and an interactive API demo, and the ClinGen Link Data Hub, and same thing for that. And if there's time, we can get into the demo. And I'd like to leave some ample time for questions and additional interactions. And so the APIs will be a little more technically oriented for developers and people who are thinking about actually integrating and using these services. But hopefully provide a general overview of these things for the non-technically inclined. And please, as questions come up, please place them in chat. And I'd like to make this as interactive as possible, especially the last 20, 30 minutes. So starting at the higher level overview of how we view defining the problem that ClinGen attempts to solve is looking at the lens through the patients, clinicians, laboratories, and researchers. And the first step is aggregating the genomic health and data and how that relates to this curation based on ClinGen's critical questions of gene disease validity, variant pathogenicity, dosage sensitivity, and clinical actionability, and how we actually disseminate that expert curated knowledge. And the end goal is improving patient care through genomic medicine. So the NIH developed the ClinGen project, the Clinical Genome Research Program, to address these growing needs, enabling clinical testing labs, researchers, patients, providers to share data associated with systematic implementations of standards for clinically relevant genome variation. And some of these things, such as genetic test reporting, variant classification, and data exchange. This involves the aggregation and sharing of genetic health data, genomic and phenotypic data, and the public domain through centralized and federated databases for clinical and research use. We'd like to curate the clinical significance of genes and variants through expert consensus, supported by scalable infrastructure. Next, we'd like to disseminate this knowledge, this derived knowledge, through the genomic knowledge to the community, through the website, programmatic access. And that's where the APIs and the streaming data come in play. And finally, I'd like to evaluate the impact of work to ensure the patient care is actually improved. So some of the key questions we're attempting to address is, are these genes associated with the disease? And if so, what is that mechanism of the variation that causes the disease? Is this variant causative in any way? And what information will affect medical management of patient health? Finally, the ClinGen effort results aim to make information publicly available and build this genomic knowledge database by improving patient care through genomic medicine. And so before we start talking about the data, it's important to talk about the software that uses that data. And why that's important is how people will actually interact with the data. And so ClinGen software has been written to both store lower level data, such as variant naming, through registration services, and higher level information, such as interpretations pertaining to variants, genes, and other topics, using additional layers of software being deployed to allow users to browse this data through user interfaces. And we're going to talk about how the user interfaces actually interact with the streaming data and the APIs as we go forward. So behind the scenes, data coordination is performed by software that leverages something called application programming interfaces, also known as APIs, which I'll refer to them going forward, and streaming services to interoperate between the different ClinGen software services. So we've worked hard to expose some of these services directly to the public to allow internal and external use of obtaining, editing, and writing to individual databases using predefined processes, rather than having to expose the databases themselves for a different type of ClinGen software. And that poses additional risks of, individual data access and people accessing the databases and accessing the servers, whereas you've actually wrapped these things in some services, APIs, for example, you can actually allow people to interact with your data in a responsible and scalable way. And so some of these categories that I'm actually going to be talking about and breaking down the different bits of software, such as variant, evidence, and aggregation. First topic, the second topic will be curation tools and supporting infrastructure. And the third topic will be knowledge dissemination. So the key question we have is, where does the data reside and how is it accessed? So first off, where does ClinGen data come from? It comes from a variety of sources, as you might suspect. So we utilize a lot of external data sources and a lot of hard work from a lot of other groups. We try to incorporate and utilize as much as possible. One, to actually leverage their work, but also expose their data to additional institutions and individuals that might be interested in it. And so the Allele Registry, and I'm going to go into some of these in some more depth, but I just want to kind of give you the breadth of some of the data that we're actually excerpted, and ingested into our services. Just as a couple of examples here. So ClinGen, the Allele Registry, we get data from ClinVar, DBSNP, Exact, Nomad, myvariant.info, Civic, PubMed, Ensemble, Main, and others. Link Data Hub has a lot of these similar overlaps, but you also have some additional data that it goes beyond the data that we actually contain in the Allele Registry, which is more associated with variant naming. ClinVar as well, and some of these other ones are actually some different things, such as evidence, some functional data. We also have the variant classification interface that also gets some of the similar data from the same sources, but also includes some additional information like MyGene.info and some additional ontology services. So you can see there's a good amount of services that we're ingesting from, and this is a subset of the list, and subset of the tools. And so we also have a lot of ClinGen generated data itself, whether it be from ClinGen members or external members that will actually contribute data through our software services, which is somewhat novel in the fact that it may leverage some of this above database information, but it actually contains some novel information that we actually want to contain, store, and disseminate. So such thing as the Allele Registry is actually users providing registration of their variants. The BCI and the GCI, variant curation interface, and the gene curation interface. So the pathogenicity calculator and the CNVO calculator, actionability curation interface, doses cure interface, community curation database, and others. And so what are these principles that we follow in regards to storing and allowing people to access ClinGen data? So we abide by these principles that they call the FAIR principles for open computing standards. And that stands for the findable, accessible, interoperable, and reusable. And so we talk about findable, we talk about unique and persistent identifiers, rich metadata, and searchable. So people can actually get what they're looking for in these predefined fields. Accessible means that people can actually get to them, not only in a nice web UI, but also additional mechanisms that will actually allow them to interoperate between their systems and our systems and vice versa. And this includes such things as REST APIs, UIs, message streams, CEPIO GA4 data standards. Making things interoperable. So just because people have data that you'd like to share, it may not be compatible with your data. So a lot of work is going into, like I mentioned, the CEPIO GA4 data standards above here, but actually standardizing some structured data to allow people to actually utilize that data. And when you say something, what does that mean? When you actually say standard ontologies, what are your intentions behind that? What is that representing? Versus just having more of the Wild West where people can kind of put whatever data in they want to. And also reusable. So we're going to track the provenance, open source, and a microservice design that allows people to use individual bits of these services in their service versus having only one portal in, one portal out. So where does the data actually reside? So as we talked about, we have these three categories that'll help us kind of think about the different bits of software, and including the variant evidence aggregation, curation tools, and supporting infrastructure, and knowledge dissemination. And if you have not already visited clinicalgenome.org, I advise you to. And I'm gonna share that page in a little bit and how it relates to the individual bits for the knowledge dissemination. So I'd like to give a small kind of an introduction to the first and the third column to let you give a little bit of background of when we actually talk about a certain bit of data, what tool is it in and what is that tool actually representing? What is the purpose of that tool? So the first thing I want to talk about is Allele Registry, and we're actually going to do I'm going to do a demo of this in a little bit. I'm going to show what the UI looks like, what the API looks like, and how people actually can leverage this. And so the URL is here up top, and when I show you the website, I'll also show you how to access all these things just from the clinicalgenome.org page. And so what we're attempting to do here, and this is a brief overview, I'm actually going to share some more details later, but what we're looking for is stable and consistent variant identification and naming for curation and pathogenicity. And this is one of the FDA requirements for classification process. We're also looking for open sharing of genetic information. We're looking to accomplish aggregation of information from different sources. Having identifiers on demand, which is high bandwidth, low latency, and I'll share a little bit of stats on how many variants we can register in a certain amount of time. We're also looking for API support for programmatic registration, whether that be getting data in from external sources, user submissions, and also access. So the way we were able to do this is supporting large batch queries and registrations versus having a lot of individual high overhead registrations. And all our data is structured in JSON output, which is it's an acronym for JavaScript Object Notation. It's a standardized format for people sharing structured data. And so that allows people to make a query to the Allele registry, get the data back that they need, and actually integrate it into their project. And the reason we're able to scale is actually stored in a binary low-level backing database, and it's also free and open registration. So I invite people to register, and I'll talk about that a little bit later. Another tool I'll talk about is the Linked Data Hub. And so briefly, and I'll have a little more details about this product as well, but it's essentially facilitating efficient access to collated information such as links and select data from different data sources. And this is also made available via REST APIs, apart from the UI itself. And the links information is by human genes and variants to support curation efforts. And the backing part of this is a Rango DB database. And I know this is some jargony stuff, but it's a NoSQL hybrid graph document-based database. What does that mean in layman's terms? So a lot of databases kind of look like an Excel spreadsheet where you have your columns and your tables types of things, whereas this actually is more of an efficient database, so NoSQL, which kind of means this non-relational database model that allows it to store documents. And so we can kind of create a document model and allow people, to upload data that fits that format. And it's a more efficient way to serve up some linked data. So the functional data repository is actually a fork of the LDH. So it's very similar in its functionality, but it's specific to being a structured framework representing allele functional impact. And so ClinGen identified the need to capture the rich allele functional impact data in the literature, which would be diverse types of functional assays and structured data in the literature. It enables deep modeling of storage and functional data. And this is currently utilized by the VCI experimental tab. The data exchange is another very important thing that we're able to use to share data in between services. And I'm going to talk a little bit more in a couple of slides about what the APIs actually are and what the data exchange is built upon, the streaming service. And it's another model. It's more of this queue-based model that allows us to share a lot of messages in a particular order, whereas the APIs are more efficient in actually responding to a certain question. And I'd like to show some of the differences as we go forward. And so this is a platform and data model and tools produced by the ClinGen Data Exchange Working Group. Aims to enable a consistent approach to produce and consume the full suite of genomic knowledge that's curated in ClinGen. And this allows internal ClinGen systems to set up topics to produce, consume, exchange data between bits and software. And I'll show some examples of that. And it may be exposed to additional groups in the future. And this is based on something called Apache Kafka. And so what does the data exchange look like? And I borrowed this from their website, dataexchange.clinicalgenome.org. Check it out. There's a lot of very good information there. And so you can see that we have all of these different curations and these different structured data coming in, something they're called the common ClinGen data model, where there'll be dosage sensitivity, actionability, gene disease validity, somatic, sequence variant interpretation, structure variant interpretation, and CAD-read. And so we get all this effort is allowed to getting everything into this data exchange, and we have these different topics that people can subscribe to. And so what that also allows us, the individual groups to do, is have this data in this format, and the website could actually consume that data and present it. And I'm going to show you an example of that in a little bit of what you actually get on the front end. And you can see that downstream, that this is where the interaction comes from these portals, whether it be APIs, the actual website, but the backing here is based on these messages that people can either produce or consume. So 2 examples. I mentioned the linked data hub earlier. So one example that we're able to do to support the variant curation interface, the VCI, is we're about to produce these custom JSON messages to support variant prioritization. And I'm not going to talk very much about this, but variant prioritization would be a function of the variant curation interface that allows people to do bulk creation essentially. And so the more data that people can come in, bring in for their initial search will actually allow them to do a lot of very, very cool filtering and allow them to whittle down an interesting data set and actually produce some bulk curations based on that data set. Versus having to do things manually. So it's going to be it's going to be an awesome tool coming forward. And so to date, and this is an alpha phase, so this is the VCI, and we'll have some more information about this going forward, but we're able to generate about 4.5 million messages for about 111 genes. And so this is, very early phase, But the very important part about this is we're able to produce these structured messages in this JSON format, submit them to the ClinGen data exchange in an LDH topic, and then the VCI actually subscribes to that topic, and they're actually able to get the data in this mechanism, versus us having to do, there's multiple approaches to do this, but versus us having to set up APIs for them to request directly. So in this case, they can actually basically ask the question of what's new. Another example of this is how the evidence repository here shown in this this logo here is able to get messages from the VCI. And so the VCI has published curations, and they're able to submit that to the ClinGen data exchange, and we're able to consume from that topic. And so this is Sepio formatted curations based on the evidence repository. And so to date, the last I checked, we had about 2,300 curations from 39 genes, 13 variant curation expert panels, and 40 conditions. So real briefly about what Apache Kafka is, for those that haven't heard about this. So it's essentially a distributed data storage that's really efficient at ingesting and serving up streaming data in real time, potentially from many data sources simultaneously. So it's a very cool way of actually sending a lot of messages. It can be stored in different topics, allow people to consume different structured data. And so it's retrievable in order, allows people to check what's new, and people can subscribe to, a lot of people can subscribe to the same topic. So if something is done once, it allows a lot of people to benefit from that same message stream and actually, figure out and being kept up to date at the same time, with the synchronization across multiple platforms. And so one example is our curation project, which is an awesome effort that's providing essentially an enhanced version of the ClinVar data that's curated for clinical use. And Heidi Rehm can provide some additional details if you're interested in that. And I do want to do a big shout out to all the work that's been done within ClinVar. And all the data and support, it's been invaluable. Be able to get all the structured data from a variety of ways, and we use APIs to get data from ClinVar, and we also get some data from bulk data downloads, and they provide a lot of different ways to get data. It's a lot of people put a lot of hard work to provide the data for us. So briefly, I'm not going to go through any of these in particular, but I do want to have these listed here, but these different links. And again, these are accessible from the clinicalgenome.org. But the VCI, the GCI, path calculator, actionability curation interface, this is curation interface, CNV calculator, and criteria specification editor. I do want to briefly mention some of these entities and knowledge dissemination as it relates to some of the data I'd like to talk about. So the first one is the ClinGen website and the gene graph. And so gene graph database, it backs the website along with public access to dosage sensitivity, gene validity, summarized actionability data. And the website is basically the interface to search gene graph. So it's kind of the same principle we talked about, and I'll talk about this a little bit about how APIs are built, or how we leverage APIs, is having a fundamental lower level system and having things that leverage that system. And it provides a lot of versatility and interoperability among different tools. And so also it leverages a ClinVar submission generator, and it uses something called a GraphQL API behind the scenes. I wanted to show the website briefly, because I'd be remiss if I were talking about data and I actually show you where a lot of the, even majority of this data resides. And so you can start here at the top. And so when I talk about the tools and the links, you can click on this. You get the links to these various software bits that I've talked about. You don't have to worry about writing down the URLs or anything. But this portal here is very powerful, and it actually leverages that GraphQL backend. It is able to get to all these things and present so much information in such a concise manner to allow people to actually browse through. So I'm just do a quick search, and do search for PAH for gene. And it's very fast. I just want to show you kind of what loads up. And I encourage everyone to check out the website and browse around. And so yes, it said found the gene here, and it shows that there's some curations associated with these different tools. So I click on that. I give you some curation summaries here, where you can actually look at the gene disease validity, dosage sensitivity, clinical actionability, and variant pathogenicity. And actually it provides links to all these entities. And so much work has been done for this, and it's a really awesome portal. And it's really nice to be able to do the very simple search and get your hands on exactly what you're looking for. It also has external genomic resources tab, and also has a link out to the ClinVar variant itself, if people are interested. You can see the ones that are actually associated with the PAH gene. So it's even a lot of nifty little shortcuts here as well. Okay. So briefly about a couple of these other tools real fast, and then I'm actually going to get into some of the nitty gritty with Allele Registry and link data hub, how it relates to the APIs and streaming. So the evidence repository utilizes curations from the variant curation expert panel that's published in the variant curation interface. And I think the last slide I'll show is actually showing how we pulled the thread and how we actually do this and consume from the data exchange. So it's basically systematically applying these evidence codes, ACMG and AMP, and allowing the VSEPs to actually curate its teams, which is very important. So it's also marking all curated evidence rapidly accessible to the public and designated a source of FDA-recognized variant classification. So it's basically the published version of all the work that's been done in the variant curation interface. And so the recognition enables use of ClinGen variant classification in place of traditional FDA clinical validation studies that normally require to establish variant pathogenicity. The CSPEC, the criteria specification registry, there's two things. One is actually the registry and the UI editor, but I do want to mention the editor that's coming down the path that's being worked on by Neetu Shah. It basically will allow VSEP affiliated members to actually have appropriate roles and permissions to create new specifications from either newly filled in templates or previously existing templates, edit existing specifications, and release specifications to update to the public. It also has planned VCI integration, display specific text in each of the VSEP, determining which criteria codes applied. And so we're very excited about this. It's going to be a very powerful tool to actually expand upon these specifications and how they're leveraged versus being more of a behind the scene process. And finally, the actionability knowledge repository. So this aims to identify human genes that, when significantly altered, confer a high risk of serious disease that could have been prevented or mitigated at the risk for known. Some of the goals are to develop standards of defining clinical actionability, nominate genes and diseases, produce evidence-based reports for nominated gene-disease pairs, making reports available and actionability scores available publicly, of course, and then this is available UI and API. Okay, so that's kind of a background on the software bits and somewhere the data resides, but how do we connect the pieces of what do we mean by actually talking about APIs and streaming data? So we have at least 10, 20 individual services that contain data from 10, 20 sources, and many contributors from ClinGen and non-ClinGen members, actually providing some of this custom data I talked about. And so the user interfaces are absolutely critical, and that can't be emphasized enough. But posing scaling problems when we attempt to do task or bulk and leverage the data behind the scenes in a scalable, robust manner. And that's basically says that I don't know if anyone's did genomic research 10, 20 years ago, but scraping data and downloading individual data was hard to do things in bulk. And so individual bits of software have different requirements, whether they be backing databases, coding languages, hosting services, make communication between the systems a lot more difficult. So we've aimed to build backwards and scale forward by engineering these solutions that have these built-in capabilities for interoperability within ClinGen ecosystem from the very beginning. And so that involves a couple of things involving APIs and streaming services. So a little more in depth about what an API is for folks that aren't familiar with it. So essentially, it's opening up of and this is a very simplified version of this, but it's opening up of applications, data, and functionality of third-party tools, external power users, and internal routines. And so the majority of ClinGen public-private UI interfaces are built on top of these APIs. So you build an API to do all of the functionality, and then the UI essentially calls those APIs and serves up what you're seeing in the web-friendly format. And so this uses a set of defined rules and requests are sort of valid or responses are generated, generally structured data like JSON, which I mentioned earlier. And it isn't a free-for-all. People can't just come in and do whatever you want to your data. You basically expose different endpoints, and I'll show what that looks like when we do the live demo on predefined mechanisms that allow people to do a very limited functionality. And so the basic flow of how an API works is you have a client application, say, using one of our websites. You click a button and it does an API request behind the scene, contacts a server database. It comes up with your API response and serves that back to the client application without you knowing about it. So here, I've underlined some of the ones that we have APIs for currently. And then if you go to the website, each individual one has some links for some documentation. There's some other ones on this list that there also have some APIs, but more behind the scenes, things that may be exposed in the future. So I'd like to talk about the ClinGen Allele Registry and give a little more in depth of what it actually does. So when I show a live demo of the API, it'll make a little more sense of what it's actually attempting to accomplish. And so here's this publication here by Peter et al. And so it attempts to link information about genetic variants here. And so the main premise here is this concept we call canonicalization. where you basically have an observe, and I'll show you examples of this in a second, but you have an observation, whether it be within a transcript or within a genomic coordinate, that may actually correspond to other genomic builds or other transcripts. And this service attempts to actually have variant naming that allows someone to say that, yes, these are more or less the same based on a similar type of alignment between the transcripts, the genomic context and the transcript context. And I have an example here below where you can see that we have for these 3 genome assemblies that we're currently supporting, GRCH 38, 37, and NCBI 36. We have these things that are different coordinates, but when you're actually in it, when the NCBI provides these mappings, it basically says that these are essentially the same. And so what does the solution provide? So we attempt to support stable and consistent variant identification of naming, the curation of pathogenicity, which is one of the FDA requirements. And then what we do is produce these CAIDs, which is canonical allele identifiers. So I'll be talking about the CAIDs going forward. And so we aim to support open sharing of genetic variant information, aggregation of information from different sources, abided by the FAIR principles that we talked about earlier, having identifiers on demand, API support for registration, and free and open user registration, which is instantaneous. And so here are some of the sources that we currently are able to consume from to initially seed the variants within the Allele registry from MyInfo, Cosmic, ClinVar, Exact, dbSNP, Nomad, and there's some others as well. And so here's kind of what this looks like. So a lot of people may observe a particular variant, whether they say, oh, I have a ClinVar ID. Let me start off with this number here. Or they say, I have a genome build, so GRCH 38, chromosome 11, this genomic coordinate, and the reference allele C, and it's observed it's a T. Or someone actually has a gene or a transcript that they found a similar observation. But what we would say is that, we can actually give it a single identifier here, CA321211, and we say that, what we're saying is this canonicalization service represents the same observed variant, whether it be genomic, what we saw in dbSNP, myvariant.info, ClinVar, Nomad, or all these different transcripts that have the same observation. We say, we can actually name this variant. And so this is what it looks like in the actual UI. And I'll show it very briefly before I get into the API talk. But you can see here is we have actually the identifier on top. We have links to those resources that were used to populate this, so people can actually go find where that original observation was made. We also, and then on the top part of the page is the genomic alleles, and the bottom part of the page is the transcript alleles, and we separate these out. And one thing I want to mention that before I forget is we also have, these main select transcripts that allow people to identify the actual preferred transcript that is basically this matched annotation between Ensemble and NCBI. And so give you an idea of the scale of where we're currently at. And so we have a little, this was a month or so ago, but a little over 1.1 billion registered canonical allele identifiers. And so when we talk about this high latent, this high throughput, low latency, and so for example, we were able to register all of Nomad 2.0 at the time in about 7 hours. So it's about registration of about 50,000 VCF entities per second. And so it looks like it's flattened out, but you can see when we get these big jumps, there's generally we incorporate another very large database that has a lot of unique entities. And as we go further and further, we'll actually probably hit a wall of having less unique entities before we actually start getting some more interesting structural variations. And so you can get an idea here for who's using this across the world. Of course, the vast majority of the US, but we see a lot of other countries. We get about 20 unique users register canonical alleles per month, about 250,000 visits to landing page a month. You can see this graph here. We've had some spikes, and then overall, we've got a pretty good trends going forward. About 14,000 UI uses a month from 100 countries, and about 1 million API uses a month from about 25 countries. So you get a decent amount of usage so far. Who's using the Allele registry? So we have a lot of various sets of databases that have adopted CAIDs, ClinVar, Civic, myvariant.info, Ensemble, a database of pathogenic variants, and other software bits we're talking about, but just some of these databases. We also have a lot of tools that use it. A lot of the ClinGen tools actually leverage CAIDs, the Link Data Hub, Variant Curation Base, Evidence Repository, Functional Data Repositories, and others. Also have a service level agreement with the National Cancer Institute. And so for data aggregation, we've also had a handful of individuals and groups actually use Allele Registry to kind of deduplicate and produce variant identifiers for their services. So all of us clinical laboratories. We also had the VIC consortium with one of the members, Alex Wagner. I'll mention him a little bit later for GA4GH. Across 6 prominent somatic cancer variant knowledge bases. Also the Australian genomics platform, Shariant. And we also know that Civic variant uses coordinates lookup match to match with the Allele Registry with their registration process. So I already mentioned this earlier, but we support 3 reference databases and all the transcripts in RepSeq and Ensemble. We update these monthly, and we also update these the main select, which is match annotations from NCBI and UML. And so briefly on this, and before we start getting into some of the details, so some of the challenges that involve variant curation and try to say what the impact of the Allele Registry is. And so for variant identity is very important. So the same variant is defined by differently in different sources. And so there's a wealth of information out there. If people are likely talking about the same thing, but they looked at a transcript 5 years ago, 4 years ago, 3 years ago, might have a different transcript version and maybe different genomic coordinates, and they may think it's actually something different. And it's definitely the case with genomic coordinates as more genome builds come on. It's very important to actually have some consistent naming for variant identity. Also, diversity of evidence sources, lack of standard ways to accumulate the evidence, tracking of provenance evidence used in interpretation, transparency and auditability, which are key FDA requirements, and alerting users, curators to content changes, which is one of our ongoing priorities. And so this is how we kind of see this process, how it's stacked up on top of each other in terms of variant pathogenicity interpretation via registration. So we see the Allele Registry as being the first step of actually getting variant naming. And then from there, you can actually link to different sources that talk about the same variant and also excerpt additional data elements. And finally, we actually do a dissemination of evidence related to that original observation. So the Allele Registry API, and so kind of just want to show some ins and the outs before I show some specific examples. We talked about some of these data sets that allow us to propagate or populate the original Allele Registry, and we update these as the new releases come out. So we have APIs that allow us to do these registrations in a high throughput bulk manner. We also allow users to do single and bulk registrations using that the free open user login. We also have individuals that'll do a single or bulk queries, and I'll show some examples of these. We also have various tools that actually use the APIs for getting additional information about the CIDs. They might have an HGVS expression. They might say, do we already have an identifier for this we can use? They say, yes, we do. It gets it in the payload and actually uses it going forward. And also have queries required for additional tools and data aggregation outside of things in ClinGen. So I want to actually go through a demo of this, and I do want to have this link here. And so this is part of the Allele Registry homepage. So if you go to reg.clinicalgenome.org, the goals of what I would like to do is I want to show a brief overview of the UI, show people where they can register for a new account, go over that, just kind of skim through the API documentation, let people know what's there, and actually show some examples of using CAIDs and HGVS as input. And if there's time, we can get a brave volunteer that wants to maybe try registering an API, or would actually like to say maybe they'd like to provide like an HGVS expression or something like that. And we can show what that looks like in the API and UI. There'll likely be time, but I do want to make a note that we have some more in-depth documentation on how, for example, we can actually, how we're able to show other people how they can actually register something like from Nomad, for example. We say Nomad has some data in BCF. Can you actually support this in bulk registration within the Allele Registry? And the answer is yes. And I showed a demo of how to do that. And so let me go ahead and open this up. Okay. So here's the homepage here. Let me make this a little bit bigger. This is what the UI looks like, and this is reg.clinicalgenome.org. And so from this drop down here, this is the quickest way to learn a little bit more about what we have. We can even start off with a simple example of searching this variant that I mentioned before. Doing a search for this, it brings you to this page. It says this was registered, and here's these link outs that we talked about earlier: genomic alleles and the transcript alleles. I do want to make mention that this structured data is also available, and this is actually what we're going to be retrieving via the API. And just, you can kind of see what this looks like and what data is in here. We basically have, there's more or less a one-to-one overlap with what you're presented in the UI with what you're actually able to get behind the scenes. A couple other things. So another common way to look at, get into the Allele registry is people actually have an HTTPS expression that they're interested in. They can query the Allele registry to say, does this already exist? And the answer is yes. It's one of our examples. So of course, it's the same one that we provided here. So it says, yes, this is already registered. And you can see that HTTPS expression that we searched for is one of many. So all these transcripts, you actually see that we find no equivalence in the other one. So if we have different, in this case, these coordinates happen to be the same 64 position, but you can see another transcripts are different. So it allows people to basically get a variant name to actually utilize this for curation and other efforts. And so the documentation for the APIs, which I'm going to be talking about, is here, API specifications. And there's a couple of things here. One is the actual PDF that I want to show briefly. And there's also some example data and some scripts. And I'm going to be leveraging this request with payload script as an example to show how you would actually post, either query at bulk or post, which are register at bulk. So briefly showing you what's within the Allele Registry API documentation. So you basically have an introduction, basically sending these requests, and these are the 3 examples that we provided on the website, which is the Bash script, Ruby script, and a Python script. Talk about authentication, error responses, these different parameters. What are the objects returned by the API calls? So you basically, for those folks who haven't leveraged an API before, it's important to know what valid requests are. You basically, you have a request that basically has some context. You'd like to say, like, I would like to submit an HTTPS to see what it returns. It may return something that's already registered, or it may not return anything. It'll return something if it's valid. And it might just return a canonical allele identifier. And I'll show you the examples of these. We also have the query and registration of alleles by their definition, annotation of the VCF files, which is that more complex example I talked about of actually registering Nomad data from their raw VCF file. These different types of queries you can do, and these external sources. And so to make sure we have enough time, oh yeah, there's one more thing I want to show was how to actually register a new account. So again, yeah, I'm not logged in again. So if you're not logged in, it'll actually allow you to create a new login, or if you forgot your information, you can use this button here. So it's simple as creating a new login on the page, just enter this information and what you actually want access to. In this case, it'll just be a Allele registry, and then you can register. And then immediately you can start registering new alleles. There's no process which we filter or anything like that. So let me go ahead and log in. And so now that I'm logged in, I'll be able to show kind of the differences if I want to register something manually or not. Okay, so on that page, I also have some examples that I would like to go through. And I'm going to show you what it looks like on the UI versus the API. Okay, and so the first thing you're talking about is doing a search for an HTTPS expression. So in this case, we're just going to do a search, and then we can also look at a registration. So this is observation on this transcript that disposition in A, the reference A has an observed G. And so what this looks like on the Allele registry page, just going to show you what it looks like. Just doing the search query, it returns this, and then we get the structured data here that we look like. But what does it look like when we actually do this API request? So curl is basically, it's a command, and it's in most kind of Unix, Linux, and even Windows even supports Ubuntu natively. So this is basically a command, and basically what we're saying is we want to do a get. It's a post or a put is different when you're actually submitting, but actually in this case we're actually just doing a get. Say, give me the content that has this allele endpoint, and again, there's additional information in the documentation. But give me this information with this allele, and I'm going to be providing an HTTPS. So copy this, and we see what it returns. Yeah. So it's not the easiest to see in this terminal, but it's a lot of data. And essentially what this data reflects, and it's kind of want to pull the thread between these two, is that same data that we actually pulled up for this allele. So if you're doing the same query, and if you start off with a transcript, you get the same information. So basically UI versus API. We can also do a search based on the CAID itself. If you're interested, let's say you have someone provide, let's say a tool or a user has a CAID and someone wants to actually strip out that data, or do a bulk collection that's from that data, or they're interested in just a small set of that data, they can do that. And let me just do kind of. So let's say they were interested is one example. Someone could say, is there anything registered in the Allele Registry for this CAID that also is in ClinVar? And they say, oh yeah, that it is. And we actually have a link to that here. We actually give you that ID, and you actually pull out these individual bits if you're interested in. Okay, let's do that same query, but in this case, I want to show you what you also can do in the API. You actually have, and again, this is all in the documentation. I don't want to go through that. It's pretty dry, but it's a lot of rich information in that. So in this case, we actually could say, I want to provide me with no fields. So I'm saying fields none, but I want external records. So instead of getting this long list of data, we can actually say, just give me the external records. So you can see that now it's just get the external records. So you go ClinVar, exact, myvariant.info, dbSNP, and Nomad. It doesn't give you all the genomic context or the transcripts. Let's say you just wanted a smaller response. We also have even more, you basically can keep drilling down to these different things. So you can say, okay, I want external records, but not from anything else. I just want to see Nomad. So if you do that query, it says it's basically a subset of this data. And so the point I'm trying to get across is that leveraging these APIs can actually allow people to do a lot of high throughput work between systems. So if the VCI wants a very specific thing from the Allele Registry, it could actually construct a smaller payload and actually get just that information back. And one more drill down from here, you say, oh, I don't want all the other stuff in Nomad. I just want the ID itself. So from there, now you get just the ID. So you can see how you actually could drill down to these different bits. Okay, so another example is looking at, so I'm going to show you, I have a couple files here. So how would someone do something in bulk? And so I just created a couple files here that have some HGS expressions in them. And so you can see what these things here are. And so the other entities we can do instead of get, we can actually do a post and actually provide this file from the command line. And just so you can see what this looks like. So I just have 3 HGVS expressions in here. And so if I want to do a post of this data, you can see this endpoint. We have alleles in plural. It was allele, now it's alleles. And basically I'm going to provide a file, but this file is an HGVS. And then it's basically a data binary just using this method. So if we do this post, we get a lot of information back. And so basically this is the full response from those 3 HGVS expressions that we talked about. Yeah, from these 3 HGVS expressions. Well, it's a lot of data, but you might just want the IDs. So let's say you have a lot of HGVS data and you just want the corresponding CAIDs. You can do that same trick we talked about before, is actually showing the fields none plus just the ID itself. So this is a lot more cleaner. So you can see how we would actually interoperate between these different tools of someone saying, I have an HGVS or I have a CAID and I just want something small back. And that's the kind of the beauty of APIs is actually we can expose these different endpoints and continually grow them. I'm going to show another example. So this is an example that has 4 in it. And so what happens when I do this post, I'm basically going to be asking just for the ID back. I'm going to say, okay, I have 4. So this 3 had all the CAIDs. The 4th one I added does not have a CAID. They say, oh, okay, that's interesting. So it's not registered. And what I did is I just made up this 4th example. I just took a 64 position. I said, what about a large deletion? Sorry, smaller deletion, up to the 100th position. So what happens when we do that? So you see that we don't have a CAID for that 4th entity, okay, which is interesting. So what can we do to actually register that? Let's say we have a lot of HGVS expressions and we actually want to register them, not just see what's back, what's already in the Allele Registry, but actually move that going forward. What we can do is actually do a post. a put of this data. It's another way of using APIs. And so where this is, just briefly show you. So if you're on Allele Registry, I talked about the where we have the documentation for API specifications. And so if you go to the scripts, this is this request with payload shell script. And so I'm just using that, just copied and pasted that and made it executionable. And so if I do this request with payload, I'll show you what this looks like briefly for anyone. So it's a very simple script, but what it does is essentially it creates, it utilizes your login, your password to create, it does a digest of that together. And then basically I was able to produce this token that it's able to send to us as part of this request. So it allows us to basically verify, associate, make sure that you're authenticated and allowed to actually register something versus just doing a post or doing other stuff, which does not require a login. So let's show you what this looks like. And so before I run that, you see that beforehand, we did this post of this for HUVS. Now we're actually gonna do this request of payload. And now it returns, all these have CAIDs. And you can see that, basically this is pretty much close to the size of the variants we have. There's 1.13 billion entities, more or less. And if we redo the post, we don't have to because we got back what we wanted to. But if I redo that post, you can see now we actually have that CAID back. Furthermore, you can actually remove that ID field because people actually might want to have all of the other information. You can see fields all there. And so for that fourth entity, we actually now have that CAID associated with this. You can see there's a lot of transcripts, so I won't go up to the top. I want to have time to go through this, but I also wanted to mention that they have this web page and it puts on the slides as well. And basically it's a step-by-step process of how to format publicly available data into a way that actually allows you to annotate or register a VCF file. And so I'm going to just briefly show that because we won't be able to go through it probably, unless at the very end, is we basically start off with the Nomad V2 leftover data in this case. So I started off with a smaller chromosome. We have the sites VCF. And what we can do is get that data, do a checksum to make sure it's possible. We basically get an assessment of how many lines we have. We can extract the headers and actually use this to format what we need. I know if you're not a programmer, some of this stuff looks a little complicated. It's actually pretty straightforward. It's just a public way of showing you how you can use some publicly available Linux or Unix commands to actually do these things pretty straightforward. And the main just here is actually just registering 100 K variants. It takes about 2 seconds if you strip out a lot of the heavy annotation data, but about 3 minutes, 3 minutes, 20 seconds if you don't. And so there's some additional information here in the Redmine project for the Allele Registry if people are interested. I don't see anything in the chat right now about questions, but we should have a little bit of time at the end to go do some more interactive as well. Okay, so I'm going to briefly go through the LDH and just show what the API is and where the REST endpoints are. So I want to make sure we actually have enough time for discussion and we can get back to some of the stuff if we're interested. And so for linked data, what is the need for a system that links data instead of directly integrating with third-party external databases? So there's no universal support for permanent references to be used as supporting evidence for variant interpretation. That's a problem. So also no versioning of evidence of any metadata that may have changed, receiving notifications and updates with past assertions or curations. And so if something changes down the road, we actually want to come up with a mechanism that allows people to say that something has changed and it may actually change their assertion or their curation. And tracking of provenance and standardization of data and data access across sources. And so this is the linked data vision is kind of where we're heading. Is we kind of what aims to be software infrastructure like PubMed for gene and variant knowledge, and we're more likely going to have a different interface on top of that that allows people to actually go in and actually look through more of the Allele Registry and linked data hub. But essentially, for these purposes, the primary subject is the CAID variant. And this is the main point I want to talk about: is that once we have the CAID variant, we can actually use the naming to link data across multiple data sources. It also produces low-level variant data to support the VCI and user-provided links in the Allele Registry, and basically provides those links that you saw in the Allele Registry plus some additional information like evidence. And so we have these two use cases. One is for this bulk curation process we talked about for the variant prioritization and the VCI, starting with some data, assembling any evidence, population frequency, seeing what else is in the linked data hub, and actually sending this along to the VP process to be consumed in the ClinGen data exchange. We're also working to get gene-specific knowledge from these sources as well as an ongoing process. And we're actively recruiting stakeholders, and we have a monthly call if anyone's interested. And actually embedding that and then getting certain excerpted information into the linked data hub, which then can be accessed by the VCI via the LDH API. This is an example of what it looks like, and I'll show this briefly in the live demo. But LDH being able to leverage the CIIDs, so you can see here, it's a little bit small, but with this entity ID, you can see that we're actually able to use this identifier and actually link this additional metadata that we found in other locations. And the way we're able to do that is that if you found population allele frequency from one source that had a certain variant observation, and then the impact statement had a different observation, but we actually look at the little registry and says, oh, we have the same identifier for that. That allows us to actually link that data together and actually serve this up in this linked data document, the JSON LD. And so we also want to follow the ClinGen CEPIO GA4 GH standards and making sure that that everything's available via REST API as well as UI, as long as it's permanent record link. It's very important so that data doesn't keep changing and people can't refer back to things that have broken links. And so briefly, the content that we have in the LDH, and here are the counts, and I'm not going to go through each one of these, but you can see that we have different things like functional impact, molecular consequence, we have different evidence, and how many genes we're actually supporting. And we aim to keep this data up to date as frequently as possible. And again, so we also have the link to the LDH up top. And so one of the use cases we talked about, so in the past, we were able to excerpt civic evidence. And so you can see an example that here, we actually have the CAID here. I know the text is small, but it's blown up a little bit larger here, along with the actual civic evidence that we were able to obtain using their APIs. And so it's kind of a nice process where we can use some other people's APIs to get data in a format for ingestion within the LDH. Also mentioned the LDH, getting ingested data in the LDH is also producing this data on the fly for variant prioritization within the variant curation interface. And so you can see some additional information here where we have just gene symbols, some data from ClinVar, some stuff from Ensemble, Nomad, LDH, stuff that already exists in Allele Registry. And we're able to produce these things that are actually hopefully be consumed for variant prioritization and allow people to do the curation in bulk. And finally, just regarding regarding external data sources, this is where we're heading right now, and we discuss in our monthly meetings. It's the first Tuesday of every month, as we've currently engaged with Bracket Exchange, Civic, Loved, MaveDB, and MCQR for mitochondrial, and actually scoping their data. And we're in the process right now of figuring out what data exists, what kind of structure that data is, and that'll actually allow us to propagate that data into the LDH. And actually allow other people to leverage that data further downstream, such as the VCI and others. And so this is essentially kind of pulling the thread from this process. And this is one example, just kind of this curation process. We talked about APIs and streaming and kind of what does it mean at the end of the day. And so we mentioned how we can get data into the Allele Registry via APIs. And then from there, we can also, registration of different data, maybe evidence data and stuff like that into the LDH via APIs, which is an ongoing process. And then from there, we actually have things that can query our services via APIs. So let's say the VCI wants to know something about a CAID and also additional data in the linked data hub or the functional data repository. We actually do queries from there. And then when some of this data is presented in the VCI to curators, the VSIPs, when these things get published, it actually gets published to the ClinGen data exchange. And then the evidence repository, which is kind of the published version of this Curations, is the actual consumer of what the data is actually going to the ClinGen data exchange. So we kind of have this process where behind the scenes, we have a lot of these things where we're talking about the APIs and streaming services that actually allow to make all of this possible. And so the higher level view, you can say that we can say, oh, the VCI talks to some other services and that gets published in e-repo, but behind the scenes, this is kind of like what's actually happening. And so one thing I mentioned about this was that, when it's sent to the data exchange, it's a CEPIO format, which is the Scientific Evidence and Provenance Information Ontology. It's transferred document. It contains published curations transmitted into the streaming services. And that's something that I'll mention in a second that we're continually working to improve upon. And so going forward, so ClinGen is an ongoing commitment towards continually improving data availability and access for users when adding, enhancing, exposing additional APIs and additional useful features. So we're constantly, getting feedback from users and also working things through our software prioritization process of things that would be also of interest to users and other tools. And we make sure there's access to additional structured data that's not yet exposed. So some of the tools that don't have APIs, or at least publicly facing, they do. So we're continually figuring out ways to, allow others to actually integrate on their end. So ClinGen is also working towards, additional interoperability with other groups. We're in close contact with the GA4GH, Global Alliance for Genomics and Health. It's adopting variant annotation, variant representation, experimental structured curations of functional data models for good interoperability between our groups. So that's a healthy relationship, and we really like moving forward with our groups. Eventually like adopting each other's standards, making sure that we get the widest possible audience. We continue to look forward to additional opportunities to expose our data standards with this efforts for the adoption of other popular models and data sets. I don't have much time yet, but I'm just going to briefly show where the LDH endpoints are and show you what those look like briefly. So this is ldh.clinicalgenome.org. And you see in the top right here, we have a link to our API docs. And so this is a nice splash page. It actually says what we have. I think one of my favorites is this service endpoint. It gives you a breakdown of the individual items that we have for these documents. And again, this is a Rango DB document model, a hybrid graph document model database. And so each individual item basically shows you where these endpoints are. And briefly, one of the things I like to do is, so you can click on any one of those. And if you just click ID, type ID at the end of these, you'll actually start giving you some of these data elements. So if people are interested, you can actually see what we have in there at the granular level. And yeah, so here are endpoints, different IDs, different types, different ways you can query based on CID or other types of identifiers, such as this population of road frequency source. In this case, is a no-man identifier. We also have these different modifiers that allow you to actually look for low-level, different levels of detail, searching kind of in bulk. You basically can come separated entities. You can actually ask for only particular fields, a certain number of pages, and you also have what we return in its response. So you can see what you can actually search for. If you want to do a certain path, or you just want to see what the ID is, or you want to see what the entity type is, you can actually do that specifically from the API. Okay, so before I conclude, next up, we do have a break in a little bit, but then there's also two main sessions. They both look fantastic. Hopefully people find something that they like. I do want to have questions. I mean, I have 12 minutes, but I'd like to field as many questions as possible. But while we're fielding questions, there's a lot of people to acknowledge. I'm not going to list everyone specifically, but a lot of people have put a lot of work across ClinGen and outside of ClinGen to make all of this possible. So with that, I'll conclude and get some questions.
Oh, I had a question.
Sure.
So one of the limitations that I've seen on the CID system is that there's a lot of there are a lot of variants that it cannot currently handle, such as like the larger deletions or duplications in this system, which can cause some issues with currently variant and or gene curation performance. Is there a plan to integrate those, or are there specific challenges that are preventing them from being in here as opposed to ClinVar?
Yeah, that's a very good question. Thank you for asking. So we do have a size limitation for CAIDs, and I didn't get into. I'm actually speaking later today at the Allele Registry, and I'll briefly mention this. We support, I guess, CNVs, and we're also working to support non-CNVs. And so basically, it's that next level of support for these entities that we don't currently support. In that case, I would say that we most likely would be allowed to categorize these things as a CNV. And in that case, there's no size scoping problems. And so right now, what we're currently supporting is, and I'll show you just a brief example of this. We're calling them CN IDs. So if you look at, so we have these HUS descriptors, and so you basically, you can see here that like we have this different type of nomenclature. It's kind of this hybrid HCVS type of descriptor, kind of similar to what ClinVar uses. I'll just do a search here, just kind of give you an example. So I'm just doing a reference sequence of position for CACN ID. I was hoping to see if we can see like a large scale deletion or something. No, it looks like some copy numbers. X0, so maybe a large deletion here, perhaps. So that is essentially our approach, is to continue to support CNVs and non-CNVs, structural variants that don't fit nicely into what we're calling smaller variants, which fits within our CAIDs.
Just to clarify, CACN IDs are in fact CA IDs. It's just that they are special type with their special descriptors and identifying information, but they're still not covering all the classes of structural variants. They may cover 80 or 90% of structural variants. We're still working on more complex variants such as fusions and such, but we do support currently this CACN ID namespace, which can allow us to handle insertion copy number variants.
Kevin?
Thanks, Alex.
Kevin, you have some questions in the chat.
Oh, I got you. I pulled it up right now. Okay. Okay, so our bulk downloads available for annotating variant calls and NGS data with ClinVar, or sorry, ClinGen variant or gene level info, like downloads from ClinGen. Sorry if I missed this. We don't currently have, that's a very good question. We don't currently have like an FTP or anything that's set up that actually has these. But what we do have, and this is hopefully clear in the API documents, is actually allowing people to do that directly via API. So I would say this is more of an API task that we allow for. So for example, someone could query the Allele Registry that says, I want, I used PAH as an example earlier, I want all variants and all data associated with PAH gene, for example. So that'd be an example of how to do a bulk data download. You could also query based on genomic coordinate. You basically ask for everything for a particular chromosome. But that's a good question though, in terms of maybe actually providing some of these in some sort of version way. I guess the reason we haven't really done that in the past is that, you know, the registry is a kind of a living, breathing thing that, you know, someone could have, by the time I started the presentation, someone could have registered 1,000 new variants, and that would have all of a sudden made us obsolete. So, but yeah, well, maybe get some thought that's maybe having like a monthly release, or at least allow something to get the vast majority of the data. Yeah, Robert, let me know if you have a follow-up question to that. Larry asked, transition size boundary between CA and CN. Yeah, it's about 10,000 is the size limitation that we have for, we say particularly for CAIDs. But Alex made a very good point that they're the same thing, but it's a slightly different scope. It's a different type of support that we're providing, providing for these different type of descriptors. Oh, I see your son. Yeah. Right. So downloads from ClinVar. Oh, okay. Yeah. Okay. I got you. For annotating variant calls and NGS data with ClinVar variant or gene level data. No, I think I answered that the correct way, even ClinGen or ClinVar. Yeah. So
you did
again,
totally. Sorry. Yeah.
API. Oh, good. No. Good.
Perfect.
No,
Thank
thanks
you.
for clarifying. Yeah. So yeah, API based, I guess, would be my answer. But very good question. Thank you.
Thanks.
Kevin, What
Yes.
is the policy regarding assigning CAIDs to the reference?
So the references, so I guess the way we handle that is we have two databases. Essentially, we have, a genomic set of databases, for individual references that way, and also, I guess, transcripts as well. So if I understand your question correctly, I mean, so the canonicalization process, it's supposed to span across everything that we essentially, that says, some sort of equivalence, in this case, it happened to be mapping provided by NCBI. And so we don't subscribe anything particularly to a particular reference, if that's what you're asking. Like a particular CAID is tied to one thing. But we have talked with Larry Babison about this concept of an originating allele, origination, basically a source, something that actually instigated that, that CAID. It's a little fuzzy in terms of multiple things can come in, different times talk about it, but at least from a given data set, making that association. Is that your question about a CAID with a particular reference?
No, obviously a reference, GRCH 38 at a given position might actually be different from GRCH 37 at that, theoretical same position. You could imagine assigning it one of those two, or both of them, a CAID, such that if you were referring to a genotype in the future, you would have two CAIDs, assuming the person was diploid there, and you would have a fully described genotype. Wouldn't you? But at the moment, generally, the reference position, the reference allele or nucleotide at a given for GRCH38 does not have a CAID. Only a variant that will sometimes have a CAID. Correct?
I can take this one, Kevin, if you allow me. Is it okay for me to take it, Kevin?
Yeah, go ahead.
Yeah, CAIDs are not meant to be reference specific. In other words, multiple reference specific variants or contextual variants, as we call them, can correspond to the same CAID. So that corresponds to the notion that even though we have new genome assemblies, people think of these new variants as being the same as the ones observed in previous assemblies. So to the extent that this is true, and we don't need to revise our complete knowledge of genetics with every new assembly, CAIDs will be meaningful.
But I think the question is normally, like, if I'm trying to enter something into the registry to say, does this variant already exist? The question is, it asks you, what's the reference allele and what's the variant allele? So I think what he's saying is, well, what if you want to know the CAID for the reference allele? So you'd have to put G to G. If you do that, does something come up?
Yes, you certainly have such things occasionally already in the allele registry, but I'm not sure I understand the policy. Do you wish to have these, or are these accidentally in there? Is there a systematic effort to annotate or give CAIDs in this way, or is it just happenstance that occasionally you will happen to have them?
Oh, you can have an allele that's the reference allele. So allele registry is neutral with respect to, what's the reference allele. It depends on the assembly, right? So one allele can be a reference allele in one assembly, but the alternate allele in the other assembly. And that's perfectly fine from a registry's point of view.
Right. So would it be okay to just submit the entire reference and say, let's just get all the CAIDs now, rather than have the occasional ones like the one being shown, be 1 out of a million that are currently there?
That's correct. You can, in fact, register as many variants as you wish They don't even need to be observed in any living human. They can be hypothetical. You can make computational predictions, for example, about hypothetical variants, and you'll get your ID and be able to store that prediction by linking the information about these variants. So that's in fact the policy. There's no requirement for an allele to be actually observed in a living human for it to be registered. Yeah.
Okay, and then, sorry, just to follow up on that, have you seen efforts to specify genotypes using CAIDs or some modification of them?
Registry was originally started with an idea providing identifiers for individual variants, not genotypes. But we do have plans to extend registration to new types of variants, call them variants. Copy number variants were the first to go, but we do have plans for haplotypes, diplotypes, and other kinds of entities. And we'll follow a similar policy like for copy number variants. There'll be CA identifiers, but they'll have a separate sub namespace. For example, immune or MHC, pharmacogenetic variants, and so on, call for new types of identifiers which are referred as variants, by different communities, but they have special characteristics. So we have actually a framework to do that, and we can talk in detail with you if you wish in a separate call. But to just give you a sense of what's involved, when we move from, say, variants in the sense of small variants to, say, copy number variants really are not variants. They're attributes of a diploid genome. It's a number of occurrences of a large segment of DNA in the genome. So they're really attributes of a diploid genome, not individual variants as the small variants are. So one way semantically to integrate these two concepts is to find the common denominator, which is, what is the minimal requirement for these identifiers to be meaningful? The minimal requirement is that given an identifier at the diploid genome, we should be able to tell whether the variant occurs in that diploid genome, right? This is the minimal common semantic denominator. And we use that concept to integrate these different namespaces. And I can give you a little more details if you wish in a separate call.
Thank you.
Great. Thanks again, everyone who attended and stuck around. I really appreciate it. I think I put my email up, but please let me know if you have any questions. Yeah. Thanks again for attending. I think we're next thing we're on is a break. Yeah. Okay. All right. Thanks again, and thanks Gene for your support as well for hosting.